Come.
Hi, everyone. Hi. Thank you for joining us today in a day that is different than the usual. Today, we welcome you to a C3C conference. Thank you for joining us to learn a little bit more about the dark side of conjugative plasmids. So even the title is suggestive and really leaves us curious about what we are going to learn today. And today we welcome as our invited speaker, Francisco Dionísio, who I thank in advance for accepting our invitation and being available to share with us what we will learn with him today. And before I give the floor to Francisco Dionísio, I'm just going to first invite you all that are attending this webinar to please leave your comments and questions during the session on the chat that you have on your right on your screen, the live chat of YouTube, because after the presentation, we will have a few minutes for Q&A, for questions and answers. And also present our invited speaker, Francisco Dionísio. He's a researcher at the Center for Ecology, Evolution, and Environmental Changes. He's also assistant professor at the Faculty of Sciences of the University of Lisbon, and he's doing research and in evolutionary ecology of microorganisms with a focus on bacteria, on virus, and plasmids, of which we are going to hear about today. His approach involves both lab work and theoretical research, and he studies bacterial genetics, cooperation, competition, spite, and sex among bacterial cells, and their impact on pathogenicity and antibiotic resistance. So without further ado, I now give the floor to Francisco Dionísio. Thank you.
Okay, thank you, Marta, for the introduction and for the invitation. So I'm going to share my screen. Okay. So the dark side of conjugative plasmids. So this work was done with 4 colleagues. So here are they, João Rebelo and Célio Domingos. They did a lot of work here. I'll tell you in a moment. They have a master's degree in bioinformatics and computational biology, and then also collaborating with Teresa and Francisca from in the same group, and they are both microbiologists, and it has been a very nice collaboration. So let's start. The role here of João and Célia was that this is mainly work based on simulations, computer simulations. So it was really a very nice input from them because they have this master degree. Okay, let's start. So first of all, what is conjugation? What is bacterial conjugation? So this is just something that bacteria do several times, and it is not. It can also be observed in yeast or some other living beings, but this is quite rare. In bacteria, is much more. It's really common. So conjugation or bacterial conjugation is just this phenomenon of transfer of a plasmid. So the plasmid is just a DNA molecule that is usually circular, and here there is a chromosome, and the plasmid likes to go from one cell to another one, and we call these plasmids the conjugative plasmids. Some, maybe around half of bacteria have these kind of plasmids, sometimes more than one type of plasmid, and the other half may have no plasmid, or sometimes they have a non-conjugative plasmid. So usually what we think is that they are plasmids, they are conjugative plasmids that just lost their gene responsible for transfer. I want to tell you something also. The genes responsible for transfer are mainly in the plasmid. So despite this name of bacterial conjugation, in fact this is mostly controlled by the plasmid. they are quite famous because they, or infamous because they carry antibiotic-resistant genes, many of them, or maybe I would say in the opposite way. So when you see some bacteria resistant to antibiotics, usually it's because they have a plasmid. It's more like that. So that's one of the reasons why we care about conjugative plasmids. So I'm here asking who cares about them. So World Health Organization cares about them because of that. So they have also other type of genes like virulence genes, and also they are responsible for multi-drug resistance in clinical.
We lost you now.
Okay. Yes.
We are not hearing you.
Okay.
Okay. So it was like the last 30 seconds.
Okay. So I'm okay. Thank you, Marta. Is okay now?
Yes.
Okay. Yes. So I was saying that plasmids are really important because they have they like to carry antibiotic-resistant genes and also virulence genes, and also they are responsible for multi-drug resistance. So they are really important because of that. Okay, they are also very funny in a way because they can do that. They transfer between bacteria from different genus or different species, different sometimes even, for example, between bacteria and plants, for example. Okay, so this is really they can do that. Of course, not all plasmids do that, but they are quite promiscuous. That's one word that sometimes we use. Okay, so let's go on. Let me. Okay. So they have, as I told you just now, plasmids often carry genes that are advantageous to the host, so to the bacterial cell. However, if there is no selective pressure for that, the plasmid can be lost. Why? Let's take an example. If you have an antibiotic-resistant gene on the plasmid, okay, it's very useful when the antibiotic is there. But if the antibiotic is not in the environment, if it is not in that place, so why there is no advantage for the host to have this antibiotic-resistant gene? Or in other words, there is no advantage for carrying the plasmid. So the problem with this is that sometimes the bacteria lose a plasmid, and also bacteria usually live in complex communities with many bacteria. Some of them may have this plasmid, some others don't have this plasmid. So what happens is that in general, cells that don't carry the plasmid grow faster than cells that carry the plasmid. So for two cells that are the same, the one that has a plasmid duplicates slightly slower. Okay, so this is, we are talking about an exponential behavior, so exponential growth. So a slight, very small difference can have a huge effect after some time. Okay, why is there this cost? Why should bacteria grow slower just because they have a plasmid? There are several reasons. People have been looking for them. So there is a biochemical cost for replicating the extra DNA, which makes sense, of course. The expression of plasmid genes themselves, and then just to give an example, a meta-analysis revealed that there is a negative relationship between bacterial fitness and the number of determinants of antibiotic-resistant genes that is in the plasmid. And also, so here we can see really the problem. So it is good to have these genes, but then if there is no antibiotic, it's not a good idea to carry these genes, or it's not a good idea to carry the plasmid. Okay, plasmids also disrupt, in fact, all the metabolism of the cell, and also I think this is a very nice also topic here, which is plasmid arrival into a new cell. What happened is that the cell, the plasmid moves with the single-stranded DNA. So what happens is that when the plasmid arrives into a new cell, it arrives as a single-stranded DNA. So this is a signal, exactly the signal for the SOS response, which means that immediately the cell stops dividing. So this is, of course, also a cost for the cell, because we are talking about the growth rate of bacteria. So if they stop growing, it's not a good idea if they are competing with other cells without the plasmid. Okay, so we have several sources of costs. And the problem is how can they survive? I mean, how can they maintain themselves in bacterial communities? Of course, one solution is if their transfer is very fast, they could maintain in a community. So let's, for example, to simplify, if you have a population of bacteria, all of them the same, similar, but some of them have the plasmid. So even if there is a cost of 10%, for example, for carrying the plasmid, if the plasmid is transferring very fast from cell to cell, leaving copies on the original cell, so if this is fast enough, it could compensate for the cost of carrying the plasmid. The problem is that several people, including myself, have been measuring the transfer rate of bacteria, and usually the transfer rate is very low, or is not high enough to compensate for the cost, or even for the what we call the segregation rate, which is a phenomenon where cells, when they divide, sometimes they don't. They divide and they forget to put at least one copy of the plasmid in each daughter cell. So this is called a segregation, and if you have a high rate of segregation, this can be a problem for the maintenance of plasmids. Okay, so the first take-home message is that conjugative plasmids are costly, and that transfer rate between cells, it seems to be too low to explain the maintenance of conjugative plasmids. There are some exceptions, and there is a lot of research about this, but I have to move on to our subject. So I have to ask again, how can plasmids maintain in bacterial communities? So one very important solution for that is the rise of compensatory mutations. So they have compensatory mutations appear in the cell after several generations. So just to give more intuition on this, consider, for example, a cell that receives a plasmid, there is a cost, for example, of 10%, which means that this cell from now on is going to duplicate 10% slower. So what's happening is that after many duplications, let's say 100, 200, or 500, it depends on the study, what we see is that some compensatory mutations appear in the chromosome, sometimes in the plasmid, but mostly in the chromosome. So compensatory mutations include genes that are encoding helicases and so on. Okay, we have here several examples. Okay, and we have there are several works published on this, and in fact, it's a common observation is that we have these mutations after at least 100 mutations. There 100 generations. There is a very nice study here published just a few months ago. No, not this one. It was 1 year ago, 2019, where they showed that there was one case where the compensatory mutations appeared in just 70 generations after transconjugations were formed. Transconjugants. I forgot to tell you that a transconjugant, this is going to be important for the rest of the seminar. Transconjugant is the name that we give to the cells that just received the plasmid, or and these descendants, so all the clones, so a cell that receives a plasmid, then it's going to duplicate, and all these cells we call transconjugates. The original cells with the plasmid we call donors, and the original cells without the plasmid we call recipients, or plasmid-free cells. Okay, so the second take-home message is that compensatory mutations have drawbacks. So the transconjugate clone typically has to replicate hundreds of generations for the compensatory mutation to appear, as I just said. And then, but for this to have, in fact, an advantage, I mean, the plasmid has to be maintained somehow. So how can we ensure in nature that the plasmid is going to be, that the selective pressure is going to be maintained for, let's say, 400 generations before the compensatory mutation appear? So how is that possible? So in nature, that may be not always the case. After a few generations, maybe the selective pressure is not anymore there, so the plasmid could be lost. There is another problem about this, is that sometimes plasmids are what you call cryptic, so they don't have any obvious gene that would be advantage to the cell. And there is still another problem, which is sometimes compensatory mutations appear, that's okay, but then the fitness of, let's say, the cost was 10%, so initially, and then there is a compensatory mutation that leads to a cost of just 5%, let's say. But a cost of 5% is also very high if we are considering that these cells are all the time, I mean, they replicate exponentially, so a cost of 5% is still huge, and so the compensatory mutation here is not is not a good solution just because of just the mutation itself. Okay, so I think this is already clear how compensatory mutations are important, but they are not enough to understand how they could be helpful for the maintenance of plasmids. Okay. One thing that we should not forget is that bacteria often live in structured habitats. So this is going to be important as in this seminar. So the fact that they live on structured habitats means that that maybe the social interaction between them can be really important. Okay, so the success of an individual when it is during social interaction is going to depend on what I call here the direct impact on its own transmission, but also the indirect impact on allele scopes present in other individuals. Okay, so the success of each allele has two parts. This second part is what we call the social interaction. It's a consequence of it's a cause of social interaction. Okay, and we have seen in the last, I would say, 20 years or 30 years that this social behavior of microorganisms, in particular viruses and bacteria, has been really, it's really important for diverse phenomena. Let's say, for example, viruses that cooperate altruistically to beat the innate host immunity. So this is a very recent work. They're very interesting, and so on. Bacterial virulence is also sometimes based on cooperation between bacteria, and so on. Okay, so the social interaction between microorganisms has been really a hot topic in the last 20, 30 years, and the more we look, the more we are convinced that they are fundamental to understand the evolution of microorganisms in general, and in this case of plasmids and conjugate and plasmids and bacteria. So there are some works that were published also a few years ago where people were showing that conjugative plasmid could be helpful to avoid the appearance of cheaters. So I explain what it means. So let's assume that we have a set of bacteria where they should produce some enzyme, public good enzymes, which is secreted. And then what's happening is that if some of the cells have a mutation and it's not producing that enzyme, this cell is going to profit from the production of the enzyme by the other cells in the group. And what is even worse is that these cells that are not producing the enzyme maybe are going to grow faster because of that. So the problem with this is that after a while, after some time, most bacteria in that group is going to be going to be mutants. So they are not going to produce this important enzyme. For example, sometimes involved in virulence and in pathogenicity. So the problem is that the important phenomenon that should occur in this case, if it is a pathogen, is no more possible. So if the gene for encoding this enzyme is in a conjugative plasmid, what's happening is that this plasmid can move to the other cells around and not only avoid these cheaters to propagate, but also to oblige them to work for the group. So to produce this enzyme that I call in general a public good. Okay. People also have shown that plasmid can be involved in what we call interference competition. I think the most famous example is bacteriocins. So they are toxins produced by bacteria precisely to compete with other bacteria around. It has been shown that they are in fact spiteful interaction. I'm going to explain what it means in a few minutes. So by secreting bacteriocins plus plasmids bearing cells, so these bacteriocins are if they are encoded in plasmid, which is very common. So when the cells produces these toxins, they are going to kill cells that are nearby that don't have this plasmid, and so they kill them. So it's this. In other words, they are killing competitors. So after a few minutes, only the cells containing this plasmid that is coding for the toxin remains in the system. Another example is that this is very similar with another example, but this time not involving plasmid, but viruses, where it was shown that cells may use their temperate virus, latent virus or lysogens, where they sometimes one of them start producing the virus in huge quantities, and then that cell is going to die, but all the kin cells are going to survive, and then the cells that don't have this virus are going to die. So in a way, these viruses behave a little bit like bacteria, since they have some different characteristics from the bacteriocin, but in a way we can look at them as if it was a biological weapon, let's say. Okay, the problem with these examples is that many plasmids may not encode toxins, nor any advantage genes. So we still have to explain how these conjugative plasmids can be maintained in nature. Okay, so and there is another problem with this, is that even if they have a very useful gene, for example, bacteriocins, what could happen is that the cell could recruit this gene from the plasmid to the chromosome, and then could get rid of the plasmid if the plasmid is costly. So So the fact that the plasmids carry useful genes is not enough to understand how they could be maintained in bacterial communities. Okay, so let's remind again, compensatory mutations take long time to appear, and only part of conjugative plasmid carry advantage genes, and they are advantaged only in a few conditions, so we have to understand how they can be maintained, why they are so common in nature. So this is still a mystery, and a lot of people is working on that. So what we thought is that maybe a solution to this problem could rely precisely on plasmid cost. Okay, so the idea is that when plasmid arrive into new cells, donor cells would be harmful to recipient cells. So this is just a way of seeing a common observation. So the plasmid transfer may be considered harmful or spiteful act performed by donor cells. So if the donor cell is happy and growing fast because it's already adapted and there is already a compensatory mutation that occurred in these cells a long time ago, when they transfer the plasmid to other cells that that never saw this plasmid, the donor cells are in fact transmitting a cost to these recipient cells, and now these transconjugates are going to duplicate slower. Could be this used by donor cells to have an advantage? Before answering to this question, let's explain what is a spiteful behavior. So here is a very nice drawing from a paper in Science. So the idea is that we have an actor and a recipient of this spiteful behavior, okay? So the actor is going to be nasty to this guy, is look unhappy. And then because of that, this is a non-relative to the actor. Because of that, a relative to the actor, which is similar to the actor, okay? So this one is going to benefit because of this cost paid by this non-relative, okay? So there is also a cost paid by the actor, okay? The definition of spite says that the actor has a cost to, and the non-relative also has a cost, and this cost is higher than that one. But we will see this later, okay? That's what I just said here in this first sentence. Okay, so what are the conditions for this spiteful behavior to evolve or to be selected? Well, according to Hamilton's rule, this is a mathematical formulation by Bill Hamilton already 50 years ago. So what he showed was that if the relatedness between individuals is a certain value, and if the benefit, or in this case a cost due to this harmful behavior is minus the cost of the actor for doing this is higher than zero, then this spiteful behavior could be selected. Okay, so I don't have time to explain here how he got this conclusion, mathematical conclusion, but we can discuss that later or after the seminar if you want. Okay, so we have here three, let me here just, so minus B is the cost that this guy is going to suffer, okay? And C, or minus C, depends on how you look to that. C is the cost suffered by the actor, and R is the relatedness between the actor and the recipient. So the relatedness between these two guys here, okay? And okay, so let's go on. A few years ago, people were trying to understand how to calculate this relatedness, and there is an important point here, is that we have to calculate relatedness according at a particular locus, not in general. Okay, so this is also more or less technical for now, but we can calculate this relatedness as of the allele. Okay, so what is the probability for the recipient individual to have this allele minus the proportion of this allele in the whole population? Okay, and here is a proportion of the allele in the actor, and here is also the same as above. So it's the proportion of or the frequency of this spiteful allele in the population as a whole. Okay, so so let me. In our case, we are going to then to understand how these numbers are, which numbers are these, and how can we calculate this. It's going to be very simple indeed. So just wait a few slides. So so to go on. Okay, so we have to now to take care of something, which is how, when we talk about relatedness here, we have to ask in relation to what? Okay, so the problem here is that when you have several individuals interacting, you can see that, even when you have just two individuals interacting, as we see here, okay, there is someone that is smiling with that, so it is good for him that the other two guys are fighting. Okay, so in a way, something he is gaining something with that. So in that case, we have to complicate a little bit Hamilton's rule and consider that the cost paid by both actor and recipient is going to can be used by the other individuals in the same neighborhood. Okay, so what I call here the economic neighborhood. David Keller in 1994, he showed that if you use this Hamilton's rule, more complex Hamilton's rule, if you substitute the relatedness here in these numbers, you can obtain a very similar mathematical expression as before, but PR and PA are in the same place as before, but PE and here is the proportion of the allele in the individuals in the same in a neighborhood and not in the population as a whole. Okay, it's just so we have to consider just in a neighborhood. So this is clearly highly important in when we talk about social behavior involving bacteria, for example, because in most cases bacteria are living in structured habitats, so the populations are structured. So this means that a given cell is not interacting with all the other cells. For example, in our colon, they are just interacting with the cells that are nearby, even if there are millions very close to it. This cell is not interacting with all the population in the colon. Okay, and that's what we are going to consider here in this seminar. So. So just to summarize, PE is the frequency of the spike allele in a neighborhood, so not in the population as a whole. This is going to be highly important, as you will see in a moment. Okay. Okay, so our hypothesis, I'm just repeating here so that we don't forget. So our hypothesis that the cost experienced by transconjugate cells, so the cells that just received the plasmid, may be advantageous to the donor cells. Okay, and so the plasmid transfer itself, this phenomenon can be considered a spiteful behavior. Let's see if this is true, and what are the conditions for this to be the case. Okay. With this mechanism, we may assume that plasmids have no relevant genes beyond the normal genes that the plasmid has to have. One is, so they have to be able to replicate inside cells. They have to be able to ensure that there is no segregation. So we have many cases like that. So most plasmids have what we call partition systems to ensure that both doctor cells receive one plasmid copy, and finally that the plasmid is able to transfer to other cells. Okay, so besides these genes, that we are not assuming that they have any other gene to maintain themselves in their host. Okay, when we used the question that we just saw to understand what are the conditions for this behavior to be selected, we find that the final expression is very simple. So here 0, I put it 0 because this was the frequency of the spiteful allele in the recipient cell, in the recipient individual. In our case, the cell that is receiving the plasmid has no plasmid. This is something that we know for a long time, which is plasmid don't move to cells where are other plasmids, the same plasmid or similar. So they don't move to these cells. They just move to cells that don't have a similar plasmid. So we know for sure that the probability or the frequency of the allele in the recipient cells is 0. And we know for sure that the cell that has a plasmid surely has this allele, which is the plasmid itself, so the frequency is 1. And P is just the frequency of plasmid bearing cells in the system. And then if you simplify this expression, this is going to be this simple expression than before. And now, if we are talking about unstructured habitat, so I put here this in red, it means that all individuals are interacting with all individuals like in a liquid medium, like if you put bacteria growing in a tube and with oscillations and so what's agitation in 37 degrees, for example, what we do all the time in the lab. So in this case, every cell can interact with any other cell in the same tube. Okay, so this means that if I put inside the tube D donors and R recipient cells, so I mean I mean, for example, 1,000 donor cells and 1,000 plasmid-free cells, then P is D over D plus R, and then all this expression simplifies to this, D over R higher than C over B. Okay, we'll see in a moment what it means. And now for what happens in the structured habitat. In a structured habitat, even if what is a structured habitat? As, for example, a petri dish. So where cells can interact only with their neighbors. They cannot interact with cells that are far away from them. So if I put in a petri dish D and R cells, and if I mix them in a petri dish, then they start growing. And what's happening is that from point to point in the petri dish, the proportion of plasmid-bearing cells is going to be a very similar formula as before, as this one here, but it's going to change from place to place in the petri dish because it's just depends on if donors or recipient cells just fall on that place in the petri dish. Okay, so in this case, the mathematical condition for the spiteful behavior is just this condition. It's very simple also, like the previous one. They are very similar in a way, but they are very different, as you are going to see in a moment. So what this mathematical expression shows is that spite evolves if the local proportion of donor and recipient cells is higher than the ratio of cos of the output to the recipient individual. Okay, so another way to say this is that plasmid donation is advantage for high density of donor cells, low density of recipient cells, and low cost of donor cells or high cost of newly formed transconjugates. So this is just several ways to see how this formula works. What is the difference between this equation and the previous one? This one is that this is the global in the total population, and here is just the local proportion that is important. Okay, as I told you, this is going to be important. So let's, now I think many people listening to this seminar is thinking now that this is very weird way of thinking on this, because when a cell receives a plasmid, this cell, we call it a transconjugant, but this cell is itself also a donor of the plasmid. So this cell could also use this plasmid as a weapon, as a biological weapon, as the original donor cell. Okay, so this is true, but what we are going to see is that, or to check for now, is how, given that the cell has to wait for several generations for this, for the compensatory mutations to appear, for some time, the donor cells are going to have an advantage for transferring the plasmid to the cells in the neighborhood. Okay, so we'll see in a moment how this happens. So, but before that, let's. What I was saying is that because of this complication that transconjugates became also donors themselves, we have to make simulations to understand the importance of this phenomenon of spiteful behavior mediated by plasmids. Because what we see here in these equations is what happens if there is an advantage at the beginning, but we don't know in the medium term. Okay, so we made simulations in unstructured habitats. What we consider here is that every cell competes for nutrients with every other cell, and conjugative plasmid may transfer from any donor cell to any recipient cell. Okay, and in these unstructured habitats, what we have to think about is in these equations. So this is going to be tricky to understand this figure, so I'm going to explain it. So first, I decided that I'm not going to talk about the figures on the right side, okay, so it would be too much. I have here, sorry, okay, I thought it was a message, but it is an old message already here in the chat. Okay, so let's see what we are measuring here. So in the simulations, we put I'm sorry. Okay, yes, sorry. So in the simulations in an unstructured environment, we just put bacteria, so as I told you here, interacting with all the others, and see where in what conditions the donors have some success. Okay. So we are calculating their fitness. I can give you the details later how we calculate that, but what is important is that if these dots are above 0, it means that donors have success in comparison with the recipient, with the plasmid-free cells. Okay, and in this case, what we see is that we have here the initial proportion of donors to recipient. 100 to 1 means that initially I have for each recipient cell, for each plasmid-free cell, I put in the system 100 plasmid-bearing cells, so donor cells. So much more donors than recipients. So it means that D over R is 100. Okay, here. So so here with this is the conjugation rate is high, is the value that we observe with the best plasmids when they transfer from cell to cell, and then this is 10-fold lower, 100-fold lower, and so on. So what we see here is that the donor cells have some success. And because this is above the line 0, and this is observed when the proportion is initial proportion is 100 or 1 to 1. Okay, so 1 to 1. And for example, just to give here an example, so this is initial cost in transconjugants was green, red, or blue it depends on how much is the cost, so different simulations. And then we have here the plasmid cost in donors and in the transconjugate after adaptation. So what we see is that if the cost is zero, then 0 over B is always 0, because B is never 0. So this is always 0. So whatever we put here, there should be always an advantage of for donor cells in these unstructured habitats. So this is what we see here. These circles here and these circles, and we can't see here, but these circles are slightly above 0. But sometimes they lose, as we see here, because there are so few plasmid-bearing cells that they are lost just by chance, because this is a simulation with a stochastic simulation. Not we didn't use differential equation. We use just a stochastic, so individual based model simulations. So they can be lost here. But we see that even if the conjugation rate is low, they have here some success here, and here, and here, and so on. But here sometimes they are lost. But when the conjugation rate is slightly not very low, they are maintained. Okay, but let's say now that C and B are 0.1 and 0.4. So 0.1 is here, is the squares of C, and 0.4 is the cost for transconjugate that just formed, and then we put the simulation running for 500 generations, and we decided that the adaptation was appearing in these cells after 200 generations. So the result, as you see here, the condition gives that D over R has to be higher than 0.25. Okay, this is 0.25. So it makes sense that here they cannot win. So this condition is the red square here that you can see here, this red square, and so on, this red square and this red square. Okay, so in all cases, as predicted by this theoretical model, very simple, but is that they should lose. But they should win above when the original D over R is 1 or 100, because 1 is above 0.25 and 100 also. So you see this square and this square here are, in fact, mean that the donors have some success here. Okay, this is what we were expecting with this our theoretical model, and it's nice that things go well. So now I explained to you how we did the structure simulation in a structural environment. So what we did here was consider that this is our cell, let's say, and then there is a neighborhood where if it is a donor cell, it can transfer a plasmid to one of these cells around it, and also they can compete with these cells for nutrients. Okay, so what we, for example, they can grow. For example, here we have a blue cell. This is a let's say a donor cell. It's going to duplicate, okay, and then also conjugation. So a donor cell and the recipient cell, so the cell receives the plasmid. So this is now a transconjugate, and so on. So after 500, a few generations, we killed almost half of the cells, and then they can regrow again and conjugate again, and so on. We do this for 500 generations. So now these are the results. So with the same conditions, just to make sure that we understand. Now this red square that was below, it was here in the liquid, in the unstructured environment. Now in structured environment, it goes up. In fact, it happens with all the other dots also, all the other cases, even when the initial proportion of the donor to recipients was very, very low, 1 over 1,000. So why this is happening? Because locally, we have sometimes this delta over rho. So what it means is that locally, the proportion of donors to recipients can be, in fact, very high. So just to compare, I put here these. So these dots, all these points are above the line 0 here, as are these points here. Let's see, before we had this. So they were all below, with the exception of the case where C is equal to 0, the circles. Okay, so in this case, we made a simulation, and we ran a simulation, and every time a donor needs, every time a donor cell donates a plasmid to a recipient cell, so it means that they have to be very close to each other in the grids. We calculated the mean value of the relatedness in this neighborhood of 7 times 7. So in this neighborhood, okay? So what we found is that there were, in this simulation, just in the first cycle, in the first 6.6 generations, we observed more than 2,000 conjugation events. Okay, so cases where a donor cell transferred a plasmid to the recipient cell, and then we calculated the relatedness at that moment in that neighborhood, and we found that the values are very high indeed. So we found that the average value is 1.8. So you should see that, in fact, initially overall in the population is 0.01, but every time there is a conjugation, in fact, the relatedness is very high, and with the confidence interval of 99.99%, it is between 1.68 and 1.97. So clearly above the 0.25, which was our theoretical limit for this behavior to be evolutionary advantages. Okay, so delta over rho, so locally the ratio of donors to recipients can be very high even when D over R is very low. So it was 1 over 100. However, when each cell is going to grow, they form colonies, and when they meet each other, they in fact have a very high relatedness. Okay, so if we calculate here, let's go back here. You see here the success of donors, okay? So if you go here and calculate not the success of donors, but the donors of untouched conjugates, okay, here there is a mistake here, okay, it's just, so this is donor untouched conjugates relative to recipients, so to cells that never saw the plasmid, okay? So when you see here, okay, the success of all the cells with the plasmid, we can see that the figure is not very different from the previous one. So these points are now higher, it's true. These ones are all higher, but all of them were already above 0. Okay, so this. Okay, I have here I forgot. I have here the same figure. Okay, just to compare. So I'm going back and forth. Okay, so this is similar, and there is this more difference here because the conjugation rate is higher. But anyway, we can see that the final conclusion is the same. So here, what we are saying is that, let me finish with this. What we are saying is that the transfer from transconjugates to the other cells is almost irrelevant to the plasmid success. That's what we are saying here. Okay, so this is really, a surprise to us that in fact the success of of donors is the same as the success of the plasmid in a way. Okay, and finally, the other conclusions are these ones. So the fitness costs frequently imposed on newly formed transconjugates is going to benefit the original population of plasmid-bearing cells even if the plasmid is also costly in the donor cells. So this is, let me just to show you. So when you see here the cost triangles and squares, it means that also the donor cells have a cost for carrying the plasmid. And even in that case, donors win in this competition with plasmid-free cells, despite having this cost. Okay, so so this advantage allows the maintenance of original donor population, even when the conjugation rate is not very high, as we saw also. So this mechanism depends on the plasmid ability to move into other cells, and not only on any particular beneficial gene, as we assumed from the beginning. Okay, so this is really a different way of seeing what's happening with conjugation. So I'm open to questions, and thank you for your attention.
Thank you very much, Francisco, for your presentation. We learned a lot about plasmids, and it is quite a complex field, and so we were able to follow a little bit more and learn a little bit more about it. If there are any questions, we don't have questions for now, but I invite those that are attending following this webinar, if you have any question, to please leave it on the chat. I would just, while we are waiting to see if we have any question, I would just leave you a challenge, desafio in Portuguese. So we learned about the dark side of plasmids, which was the title of the presentation. If you would have to summarize for someone who just came in, and what were the three main aspects which you would emphasize as three main aspects of the dark side of plasmids? I know it's a challenge.
Okay, is that plasmids are really, so the people always saw that the fact that the plasmids are costly is a bad thing for bacteria to carry them. What we are showing is that this cost can be very useful for the donor cells. So this is really a completely different way of seeing things. Instead of three things, I'm saying just one, because I think this is the most important.
Okay, it was even more efficient.
Sorry?
It was even more efficient, just more one take-home message. Yes.
Yes. So because all the problem is to explain plasmid maintenance, knowing that they are usually costly. So what we are saying is that this cost is precisely the necessary ingredient for the success of plasmid-bearing cells. Okay.
Mm-hmm. Okay. Thank you, Francisco. For now, we don't have questions on our live chat. I would suggest, because we are also approaching the end of this session, that if there are questions that arise afterwards, people can also contact you through email, right? So that they can afterwards send you an email with any question that arises afterwards. Okay. So if there are no further questions, I would now wrap up our C3C conference, starting by thanking you again, Francisco, for your availability and for your great presentation, and congratulations for your work. And thanking Mariana, who is here taking care of the backstage and ensuring that everything goes well with the connection between Zoom and YouTube. And thanking all that's attended the webinar. Thank you for your presence. And I suggest, I invite you to stay tuned to the events that we will have next week. We will actually have several webinars also next week about the Plants Fascination Day, about the World Bee Day. So please stay tuned, check our website, and check all the events that we will have next week. So thank you very much.